Hi. Good morning, everyone. Yeah, thanks for that introduction, Lucy. And as you said, it's a good thing that Katya manages to get most of those birds back to where they belong. But the ones that don't make it normally get phone calls at strange hours. And we had a shared MSc student. And the reason why I'm presenting for her now this morning is that she's no longer working at the University of Pretoria, or she's no longer a student, should I say, and has other commitments and so forth. What I have done, it's not always easy to present your students' work because you haven't spent as much time with it, but I did try to put together her summary and conclusions with some images, and I pre-recorded the presentation. So I'm just going to play that video, and then I'll be open up for some questions afterwards. I tried not to go into too much detail as far as histology and anatomy is concerned. So I'm just looking for my, I'm looking for my video. Don't know why it's not coming up. Show all windows.
No worries, take your time.
I wonder if it won't play a video. Okay, let me try. If I play my, it might work this way if I just play the PowerPoint. Is the volume okay?
I think so. No, Martina, there's no volume coming through.
If I can, hi everyone. If I can just make a comment, when you're sharing your screen, there should be an option to share your laptop sound as well.
Okay, yeah, let me try that.
Thanks, Kyle.
Sorry. Yeah, I thought doing this might make it easier. Now, how do I get out of this presentation? Sorry, now I'm I can't get out of my presentation now. I am very unfamiliar with this platform.
Okay. No problem, Martina. Escape or right click and take your time. We do want today to be chilled, and so don't feel any pressure.
No, I don't feel pressure. I just feel stupid. Okay, let's try that again.
Good morning. This morning I'm presenting to you work from a combined student from both myself and Katja Kuppel, Anel Nordia. She completed her MSc on the morphology of the Southern Ground-Hornbill gastro-intestinal tract. Now I'm sure you all know about the Southern Ground-Hornbill, so I don't want to spend too much time here. But it is an obligatory carnivore, and therefore the GIT may display specific adaptations which could be of significance in the husbandry of this endangered species. Factors that contribute to unnatural mortality in southern ground hornbills in South Africa are poisoning, both primary and secondary, electrocution on transformer boxes, shooting, and illegal capture for trade. An undescribed threat is lead contamination of hunting offal from the use of lead-based ammunition as well as object ingestion, as the southern ground-hornbill is a non-discriminate eater in the wild as well as in captivity. Carnivorous bird species have important gastrointestinal differences in anatomy, physiology, nutrition, and disease etiologies that greatly affect the success of medical and surgical intervention during gastrointestinal diseases. And here's a few other points about the southern ground-hornbill, but as I mentioned, for the materials and methods, 9 adult southern ground-hornbills of either sex which succumb to natural causes, unknown causes, attack from other birds, or euthanasia for humane reasons such as untreatable conditions and management reasons, which is usually reproductive failure, were used in this study. Deceased birds were from a Buller Ground Hornbill project and were transported to the Faculty of Veterinary Science for post-mortem examination. After the examination, the GIT with the liver, pancreas and spleen was removed and immersion fixed in 10% neutral buffered formalin. In some instances, not all parts of the digestive tract were included in the study due to obvious pathology. The topography of the organs was described and captured during post-mortem examinations. For the histology portion, after the gross description, samples for histology from the various sections of the GIT were taken, dehydrated, and thereafter processed routinely for light microscopy. The stains used were hematoxin and eosin, periodic acid shift, and Mason's trichrome. Here you can see H&E, PAS, and Mason's trichrome. The tissue samples were manually embedded in paraffin wax and plastic molds, and the sections were cut at 3 to 5 micrometers and stained with these stains. This slide just shows where the various samples were taken for histological analysis, and we'll go through the various areas in the following slides. This is an image from the bird at post-mortem before fixation, and this is a labeled image post-fixation with formaldehyde. The most obvious organ from the ventral view was the ventriculus, situated between the 2 liver lobes. There's the ventriculus in the right and the left liver lobe. The size and position of digestive organs are important when evaluating topography and pathology on diagnostic imaging. Abdominal masses can cause changes in the location of the GIT, and an abdominal mass, for instance, could even be a distended gizzard. A ventral midline approach is used in laparoscopy, and sutures should be applied to the ventriculus rather than the proventriculus, because the ventriculus has stronger suture holding abilities, and as you can see from this image, the proventriculus is not easily accessible. Additionally, most foreign bodies are also situated in the ventriculus. The liver was relatively larger than expected for carnivorous species, and it had a smaller left lobe in comparison to the right lobe. A larger liver for the southern ground-hornbill supports a high metabolic rate, protein, acid, and bile salt synthesis, and excretion and detoxification processes necessary for a carnivorous diet. The proventriculus was small and the ventriculus was large, and here you can see the comparison between the two. Low longitudinal folds were present in the luminal surface, which allowed for distension and for large prey to move through. Deep proventricular glands were visible macroscopically in the wall of the proventriculus and formed the main feature on the cut surface. Here you can see the openings of the glands. Some of the glands emptied into the cranial aspect of the ventriculus and were more concentrated on the dorsal aspect. This being the dorsal aspect. Sorry, this is the dorsal aspect here. The proventriculus was separated from the ventriculus by the isthmus gastris. That's a constriction or narrowing over here. The primary function of the proventriculus is to secrete mucus, hydrochloric acid, and pepsinogen into the digestive compartments. The viscoelastic mucus gel cover lubricates and protects the stomach surface from corrosive actions, providing a physical barrier with low permeability to pepsin and other macromolecules. The gland-free regions over here may make the organ more distensible. Histologically, the proventriculus was composed of the typical 6 layers of the digestive tract. The lamina epithelialis, shown over here, was characterized by folds, which was lined by a simple columnar epithelium. This is a low magnification image, and you can see these folds. Superficial proventricular glands, demonstrated over here, were composed of columnar cells secreting mucus into the lumen of the proventriculus. So these are the columnar cells over here and over there, and these glands secrete mucus into the lumen. The lamina propria, this area over here, contained infiltrations of internodular lymphoid tissue, demonstrating an important immune function. The lamina muscularis mucosae housed the massive, deep, compound tubular proventricular glands, which are demonstrated in high magnification over here. The glands were lined by 2 distinct epithelia, a simple cuboidal over here and a columnar epithelium over here. The columnar epithelium lined the ducts, secreting mucus, as seen over here in this image, into the tertiary, secondary, and primary ducts of the proventricular glands. The cuboidal epithelium represented the oxyntopeptic cells and secreted pepsin and hydrochloric acid, which are both for digestion. Ventriculus, or the gizzard. The large ventriculus displayed prominent longitudinal folds, as you can see here on the cut surface, and they were on the dorsal surface and ran perpendicular to the ventricular duodenal opening on the luminal surface and circumferentially around the centrum tendinium. The pylorus was on the right lateral aspect of the ventriculus. The ventriculus is highly distensible and functions mainly as a storage organ. The presence of the isthmus gastris seen over here makes foreign body removal by endoscopy less feasible, and that is due to the constriction over here. The ventriculus of the Southern Ground-Hornbill is relatively featureless as seen in other carnivorous species and contained non-food items, supporting the statement that they are indiscriminate eaters. Objects such as rocks that assist in grinding were absent from the ventriculi of the Southern Ground-Hornbills in the study. Histologically, the ventriculus lacked a lamina muscularis mucosa. So here is the tunica muscularis and the propria submucosa. The tunica muscularis of the ventriculus was thin and relatively poorly muscled and lacked the opposing pairs of the thick and thin muscles that are responsible for the contractions that grind the food in granivorous birds, such as the chicken. The Southern Ground-Hornbill uncommonly produces a casting and likely functions similar to the falconiforms that digest bones of their prey and often eat more than one meal before casting. The osprey produces pellets that are small and rarely seen, which indicates that most of their dietary items pass through the entire gastro-intestinal tract. The Southern Ground-Hornbill also rarely produce a cast. The relatively thinner wall and absence of rocks in the ventriculus of the Southern Ground-Hornbill in the present study support the notion that the main digestive mechanism occurs by enzymes and not grinding by a thick muscular ventriculus. So although over here it may look thick, this was the total thickness of the tunica muscularis, and if you'd compare it to a granivorous bird, you would notice that this layer is absolutely massive. The ventriculus coilin layer forms part of two secretory phases and is continuously worn down and replaced. Strands of smooth muscle run perpendicular to the glands that may assist in pulling the glands closer to the muscle layer, having the effect of emptying the gland contents. The thin musculature of the ventriculus would conceivably allow more distensibility, making it easy to house large prey. The cuticle, which is produced by mucosal glands of the ventriculus, function primarily to protect the mucosa from the strong digestive enzymes produced by the proventriculus. The massive deep proventricular glands, thin-walled ventriculus, thick coilin layer, as demonstrated here, and uncommon casting further support enzymatic digestion over the grinding of ingestion. The layered coilin secretion, which is demonstrated quite clearly here in this PAS stain, may assist in the flexibility of the ventriculus to house large prey. The intestine. The intestine consists of the duodenum, seen over here, the jejunum, the ileum, and lastly the large intestine, also known as the rectum. The small intestine consisted of 5 loops. You can see here is loop 1 with the duodenum. There's loop 2, loop 3, which is mostly ileum, and loop 4, ileum, and there's the rectum. The rectum was short and straight, and it was slightly larger in diameter than the ileum. CK were not observed in any of the specimens. The functions of CK in birds include water absorption, non-protein nitrogen absorption, digestion of carbohydrates and protein, microbial decomposition of cellulose, and microbial synthesis and absorption of vitamins. The absence of CK in the southern ground-hornbill would indicate that the latter functions take place elsewhere in the intestinal tract. The intestine had no circular folds. These are common structures in the mammal gut, however, it was not observed in the southern ground-hornbill. The intestinal villi, demonstrated over here, became shorter. So this is the duodenum, jejunum, ileum, and rectum. They became shorter, broader, and the tunica muscularis was less well developed. Tunica muscularis over here, and you can see it gets less well developed, and the goblet cells more numerous, moving from the duodenum aborally towards the rectum. The goblet cells secreted mucus into the lumen of the intestine, and you can just make them out over here, these little white gaps almost that you see in the epithelium. The inner longitudinal and outer circular layer of the tunica muscularis mucosa was present in the small intestine, indicating good motor control over the lamina epithelialis. So the lamina muscularis is not shown in much detail here, but it had 2 different layers to it, and that layer of muscle controls the lamina epithelialis here with the villi on. The decreased length of the intestinal villi, less well-developed musculature towards the rectum, and an increased amount of goblet cells towards the rectum support a more finely controlled motility in the small intestine and increased lubrication towards the rectum. Most water and nutrients are absorbed in the small intestine, and more lubrication may be required for expulsion of more solid digester as it passes along the rectum. The 2 layers of the muscularis mucosa in the duodenum, demonstrated here, consist of an inner circular, seen here, and an outer longitudinal muscle layer, which are responsible for the mixing of digester and propelling it through the digestive tract. The compartmentalized smooth muscle bundles making up the tunica muscularis, demonstrated here, there you can see the very clear compartmentalizations, in the southern ground hornbill would also provide more fine control of peristalsis. Over here you can see the lamina muscularis, the inner circular and the outer longitudinal layer. The small intestine mainly functions to absorb and digest nutrients. The zigzag formation, demonstrated slightly over here, was seen in the rectum together with the presence of more goblet cells. This supports the notion of the absorptive function of the large intestine. This is the large intestine here, and this isn't the best slide to demonstrate it, but essentially the villi moved in a zigzag sort of pattern and interlocked. The villi were closely packed and interlocked, suggesting that they could act like valves, opening and closing the lumen to selectively absorb or be exposed to ingesta. And this was from the duodenum through to the rectum. Sorry, these images are all rectum. So from from the duodenum to the rectum, these zigzag formation of the villi was evident, but it was quite prominent in the rectum. The villi became shorter and broader, goblet cells became more numerous, and the lamina muscularis became less well developed when moving aborally towards the rectum. So this lamina muscularis over here is not as well developed in the rectum as it was in the small intestine. The well-developed lamina muscularis mucosa, numerous goblet cells, fast gut transit time, short gastrointestinal tract, and the presence of interlocking microvilli and villi all indicate that fine control and increased absorption of water and nutrients, and emphasizes the importance of maintaining intestinal motility, making gastrointestinal stasis a potentially life-threatening condition. So, in conclusion, the histological findings noted in this study suggest that finely controlled movement, efficient absorption, and the rapid transit of digester through the gastrointestinal tract would be required to support the constant foraging behavior of the southern ground hornbill. There is a heavy reliance on enzymatic digestion, which is important in husbandry and stress-related ulceration. Stress slows gastric emptying and ion exchange, causes colonic mucin depletion and increased intestinal permeability, leading to the passage of antigens to the lamina propria and bacterial translocation. Bacterial infiltrations due to gastrointestinal stasis is life-threatening in the southern ground hornbill, and possible preventions should be considered. Stress poses a major risk for wild bird species in captivity and the hospital setting. Stress or overexposure to stress can cause physiological problems such as weight loss, changes to the immune system, and decreased reproductive capacity. Widespread empirical evidence across various vertebrate taxa shows that handling wildlife generally induces a severe stress response, resulting in increased stress hormones. It is inevitable that when wild birds such as the southern ground hornbill are treated in hospitals, they will experience stress. So in essence, the digestive tract of these birds is really honed to their lifestyle of constant foraging and very rapid digestion. And stress for these birds could be fatal based on gut stasis, or based on the fact that it causes gut stasis. Thank you very much for your attention.
Thanks very much, Martina. Are there any questions for Martina?
Stop share. Sorry if you heard the dog barking. The the last year to bark every time I'm busy.
Okay, thanks very much, Martina. I don't see any hands being raised. Let me just do a final check. What's really cool with us now having this sort of basic information going forward is it's going to help us understand things like why ground hornbills are so much more susceptible to lead poisoning, say, than vultures. And these are all the small clinical details that are going to help us figure out how to make much better conservation management decisions on the ground. So thanks very much for that, Martina. Next up, we have Natasha. Natasha, are you there?